Click for IB Biology. Here we're looking at the general principles of respiration, the making of ATP, some of the aspects of the energetics and the major stages within the process. A PDF of this presentation can be found in the description below, along with a quiz where you can test your understandings. So if asked to define what respiration is, the reply should be the synthesis of ATP, making ATP. ATP is the molecule which drives all the active processes in biological reactions. There are, of course, a good number of passive processes, such as diffusion across a cell membrane, but here we're looking at how are the active processes provided with energy, and the answer to that is in the molecule ATP. So ATP is responsible for driving many of the life processes, such as the building of molecules, the catabolism of molecules, the operation of nerves, very many processes. A small point, but an important point, is down here, is that ATP is made within a cell, and it is used within the cell. There's never a transport of ATP between cells. So if a cell requires a good deal of ATP, such as a muscle cell, it's necessary for that to have the apparatus so that it can synthesize as much ATP as it requires. Here's the molecule itself, and the chemical details of this are not required by the examination process. This structure here, this nitrogenous base structure, is adenine. And this sugar here is the ribose sugar, and together these are called adenosine. Out to the left, coming off the sugar, we have 1, 2, and 3 phosphate groups, hence the name ATP, adenosine triphosphate. Now the important bond is the one between the second and third phosphates, and we should describe this as an energy-rich bond, one which is able to be made or broken fairly readily, enabling the transfer of energy through the cell to processes, to active processes. If we want to add the third phosphate to the second one, this is an endergonic reaction. Free energy is required to be added to the process. But when the bond is broken, this is an exergonic reaction, and free energy is released from the process, and that free energy would be used in order to activate the biological process in question. An alternative view of this is to think of the formation of ATP as a reduction reaction and the breakdown of ATP as an oxidation reaction. So free energy coming out, and in this case free energy having to go in. Approximately it's about 30 kilojoules per mole, the free energy required to do so. As we go through this study of ATP, we'll discover that there are different stages in the synthesis of ATP. Classically, respiration is taught beginning with the breakdown of glucose in a cytoplasm-based reaction called glycolysis, which is a linear metabolic pathway. There are alternative ways in from lipids and proteins, and these will be dealt with in a later video. The second stage, so that's stage 1 there. Second stage is here, and this is classically called the link reaction, which is connecting the events in the cytoplasm to events in the mitochondria. And that leads us to the third process, which is here. So, link reaction would be 2. So glycolysis 1 leads to the link reaction 2, leads to number 3, which is the Krebs cycle, named after Hans Krebs. And then in turn, this leads us to the fourth process, which is called the electron transfer chain, and chemiosmotic theory, which was first proposed by Peter Mitchell. And then finally, in step 5, we will look at ATP synthase, and a process called rotational catalysis, first proposed in 1997 by Boyer and Walker. And interestingly, Hans Krebs, Peter Mitchell, Boyer and Walker all received Nobel Prizes for their work in this fundamentally important process of respiration, the synthesis of ATP. Links to the next video in the sequence are beginning to show below, along with a subscribe button and a link back to the playlist.